Hey everyone, the topic is organ of hearing in vertebrates. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. As we know, ear is a delicate organ located in the temporal region of the skull. It is also called membranous labyrinth, which evolved in primitive vertebrates for maintenance of balance and posture. Hearing function of this organ evolved for the first time in endurance when they started living in terrestrial environment where sound waves travel faster and to longer distances. A typical membranous labyrinth consists of 3 canals and 3 sacs. The 3 canals are attached at right angle to each other and to the sacs in anterior, posterior and horizontal positions. The 3 sacs, which are also collectively called vestibule, are utriculus, sacculus and lagina, each one of which possesses a depression inside with a sensory organ called macula that detects the position of the body in relation to gravity or static sense. Each canal ends in an ampulla that also carries a sense organ called crista, which is responsible for detection of the movement of head in any direction or kinetic sense. Both these senses together maintain the balance and posture of the animal during its activity. Endolymph fills the membranous labyrinth and is secreted and drained in meninges. Perilymph surrounds the membranous labyrinth for protection. Macula is the sensory organ of sacs. Its sensory cells also have hairs on the top and nerve at the base, but the cells are of two types, namely cylindrical cells and flask-shaped cell which bear stereocilia and kinocilia embedded in a gelatinous mass. A paper-thin otolith made of calcium carbonate and proteins floats over the cilia and its position can be disturbed by the animal bending or tilting in any direction in relation to gravity, disturbing the cilia in the process and generating a nerve impulse that informs the brain of static sense. When we examine the comparative anatomy of hearing organ in vertebrate, first we can see the hearing organ in cyclostomes, how it modified. Cyclostomes have degenerated membranous labyrinth due to their parasitic mode of life. Petromycin has only 2 semicircular canals, namely the anterior and posterior, and only 2 sacs, utriculus and sacculus. Lagina is absent. As they have no paired fin, lampreys and hagfishes do not seem to have three-dimensional sense of things. In hagfishes, the anterior and posterior canals also fuse together to make a ring-like structure. Sacculus and utriculus also fuse together to form a single sac. Fishes use membranous labyrinth for balance and posture only since the middle and external ears are absent, but some fishes can perceive waterborne sounds to some extent. For example, in fishes of the family Ostariophysi, 4 pieces of Weberian ossicles, Scaphium, Claustrum, Intercalcarium and Tripus connect the swim bladder with membranous labyrinth and transport sound vibrations for hearing. Fish membranous labyrinth is well developed with 3 canal and 3 sacs and crista and macula well formed. In Elasmobranch, rhesus utriculus is attached on the lower side of utriculus. Macula of utriculus is sometimes called pars neglecta in fishes. In amphibian classes, Urodila and Apoda do not have external and middle ear, and consequently inner ear is meant for maintenance of balance and posture only. Hearing power evolved in endurance by the modification of lagina, which produces an organ of reception of sound waves called basilar papilla. On the surface, on either side of head, a tympanic membrane is stretched over a ring of cartilage called annulus tympanicus, which is a modification of pterygochondrite cartilage of the first visceral arch. Sound vibrations received by this membrane are transmitted to basilar papilla through a columella bone, which is a modification of hyomandibular cartilage of the hyoid arch. Thus, in frog, a mechanism has evolved to receive sound vibrations from outside and transmit them to the sense organ of lagina so that the nerve impulse can convey information about the sound to brain. The organ of hearing in reptiles and birds, lagina elongates and basilar papilla elaborated to form the organ of corti, which is much more sensitive to perceive sound vibrations. Columella bone is called stapes, which is further elongated by the addition of extra stapedial cartilage. For better protection, tympanic membrane sinks into the temporal bone, producing an auditory canal through which sound vibration travel from outside to the tympanic membrane. In crocodiles, lagina is coiled like cochlea of mammals. In reptiles, both eustachian tubes join together to open into pharynx by a single opening. In snakes, there are no external and middle ear cavities, and hence sound vibration travel through ground to the lower jaw and from there to quadrate bone to columella, which carries them to lagina of inner ear for perception of hearing. Therefore, snakes cannot hear sounds traveling through air. Birds have essentially similar mechanism of hearing as do the reptiles, except that ear is much more sensitive in birds due to elongation of lagina. Among all the vertebrates, mammal possess the best developed hearing mechanism. Lagina elongates to form a spirally coiled cochlea. In middle ear cavity, instead of one bone, mammals have three ear ossicles, namely incus, a derivative of pterygoquadrate, malleus, a derivative of articular or Meckel's cartilage, and stapes derived from hyomandibular, which transmit sound vibrations from tympanic membrane to the fenestra ovalis that lead to scala vestibuli of cochlea. Mammals also have various sizes of ear pinna to collect some sound waves and direct them to the auditory canal. See, when we examine the structure of cochlea, it is a specialized and highly sensitive sense organ of hearing. Its cross-section reveals that three long chambers, namely scala vestibuli, scala media, and scala tympani. The middle chamber is filled with endolimb, while the other two are filled with perilimb. The organ of corti is attached to the basilar membrane and carries sensory hair cells and supporting cells and the cochlear nerve at the base. A tectorial membrane floats in the middle of scala media and touches the hairs of sensory cells when it vibrates by the sound vibrations, resulting in the generation of a nerve impulse that travels via the cochlear nerve to the brain. Here is the mechanism of hearing. Base of cochlea is more sensitive to high frequency vibration, while the apical portion can detect very faint vibrations. Sound waves from stapes enter scala vestibuli through fenestra ovalis or oval window, then travel to the entire length of cochlea and return back via scala tympani and escape to the middle ear cavity through fenestra rotunda or round window. Human ear is capable of hearing sound waves ranging from 20 to 20,000 Hz, but different mammals possess capacity to hear sounds of different frequencies. For instance, whales, dolphins, and porpoises can hear ultrasonic sounds of up to 150,000 Hz, and elephants can also hear infrasonic sounds of 10 to 14 Hz. I hope you could understand about the comparative anatomy of hearing organ in vertebrates. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.